coloration and poison frogs, as usual. Anyways, so I don't really have to convince anyone here that to understand all the diversity of life that evolutionary biologists try to study, traits that appear in multiple places of a phylogeny are very useful because they provide some sort of replication for the occurrence of phenotype. And these replicated instances of traits are usually thought to occur at the genetic level by one or a combination of I guess we could call them coalescent processes, where you either have true independent de novo mutations or convergent evolution, or where standing variants get selected into different branches due to different selective processes or even neutrality, or where species or lineages through gene flow can share the alleles coding for different traits, and you see the same patterns that we usually call convergent evolution. Another instance of replication or variation in the occurrence of phenotypes that really helps us understand how they evolve is geographic replication, right? Different phenotypes occur at different places, presumably because they may represent adaptations to different conditions. For example, in these so-called leapfrog distributions where we get similar phenotypes separated by not so similar phenotypes in space. In cases where we have both phylogenetic and geographic replication, we have very nice opportunities to really study how a certain phenotype and why a certain phenotype evolves. And I studied this in these nice poison frogs, which are the actual poison dart frogs used to poison darts. I can talk about this forever, but maybe not right now. And there are basically, or I at least categorize these species in two phenotypes, dark with brightly colored stripe and completely yellow. You can see the two yellow species are disjunctly distributed, separated by this striped, more widely distributed species. It does look as if they were peripheral, but these are mountains where this species can't occur. I've had that comment made a bunch of times. And the really cool thing about the system too is that developmentally, both the striped and the yellow species will acquire a stripe at metamorphosis. Most frogs actually acquire their patterns right when they're leaving the water. But the yellow ones sort of keep on going and lose a stripe until they become yellow. This happens in both species, and so was used by early taxonomists to make the case that these are sister taxa. It was a great example of a synechomorphy that was, ontogenetically the same and everything. However, now we know not only that the yellow species are not too closely related, but that there are a bunch of other cryptic lineages that exist, especially in the southern part of this genus in Colombia. I'm not going to go into a lot of detail here because I have spoken about this a lot. It will be in bioarchives soon, I swear. But basically, we find multiple different lineages that are geographically grouped, that occur both in mitochondrial and genome-wide data sets. And if we look at the phylogenetic relationships between these lineages or groups, we can actually see that it differs a lot from what was originally thought. We find that every yellow lineage is sister to a striped lineage. So we basically have geographically separated, phylogenetically recurrent origins of these yellow morphs, which is a great system to understand why we have these very brightly colored yellow frogs coming up multiple times, both in phylogeny and geography. And I'm especially interested in understanding at the genetic level how this is working, right? Is this truly de novo convergent evolution, or are there some alleles that are getting either transferred or just selected over and over again? And so a first step to look into this was really figuring out what gene flow is doing in terms of the geographic and historical replication of yellow coloration. How much gene flow is there between the distant yellow populations, the central striped ones, the yellow and the striped ones? And so for this, we used a recently developed method called space mix, which basically takes the spatial coordinates and the genetic covariances between populations and gives you their locations if isolation by distance, mutual isolation by distance were at equilibrium, right? So if we find populations that are very close, they are very genetically similar. If they're closer than geography says, then they are very genetically similar. A cool thing about this method is it also allows for some of the alleles in a population to be derived from a distant place, and so they won't really influence that population's location, and you can estimate the proportion of these alleles in a population. So now we can actually compare sort of short-range gene flow with long-distance migration that could result in introgression. And so what we find is a lot of overlap, as you can see here, especially along this river drainage. This is called the San Juan River. We find that a lot of the populations are sort of matched together way, way closer than what geography says. Even this population of striped frogs at the mouth of the San Juan is significantly moved towards the rest. So it's interesting that populations seem to be sharing a lot of genetic variation, especially at short distances. It is also interesting that a lot of these are not sister taxa, at least in our phylogeny, but they are close to one another. So it seems like nearby non-sister taxa are sharing genetic information. If we look at this W parameter, which tells us the proportion of alleles coming from far away, we can see these are extremely low. They are very close to zero, and we actually implemented a way to test this fully varying model versus one where we set all these W's to zero. And the base factor comparison tells us it is 1,700 times better to set these parameters to zero. So I think that's pretty strong evidence that there is very little long distance migration in this system. So next we wanted to see where this gene flow is actually happening, right? We saw there was a lot of clumping around this river, but it could also have to do with just the geographic coordinates that we gave the program. So we used this method called dyneins, which was developed recently as well, which doesn't take populations as the focal thing to analyze, but pieces of a map. And again, based on isolation by distance, identifies regions of the map where there is more or less gene flow than expected. Their example when the authors introduced this method were African elephants, and they find a very strong barrier around the jungle in Central Africa, which separates savanna and jungle elephants. We've known that is the case for a very long time. When we run it in the frogs, we find, I think, a really neat result, which is that we find a very clear corridor of gene flow along the San Juan River. Bear in mind that this program has no idea about the landscape. So the fact that it recapitulates perfectly a river, I think, is remarkable in telling us that this river is a corridor of gene flow. And it really coincides with our previous results using space mix, in telling us that the populations along here are sharing a lot of alleles. Interestingly, it also finds that this population of plain yellow frogs is actually separated from everyone else, which makes sense because it is a high elevation species, and this is basically the lowlands. It also tells us that it has a strong barrier with what was thought to be the same species but further north. I think that's interesting, considering also that this species seems to be sharing a lot of alleles with the center. And if you look more closely at this corridor, which I have been sampling for a long time, you can see that there is actually sort of a gradient from completely yellow to striped. This is the one place in these frogs distribution where you don't see this dichotomy of color patterns that I've been describing. And in the future I will know a lot more about this, but I'm currently working on it. But for now, this suggests that maybe this corridor of gene flow does involve the movement of alleles involved in coloration. And so it is probably influencing what we see in the phylogeny as convergent evolution here. So we seem to be finding gene flow, especially along the center of this, schematic that I've put up, both between striped and yellow populations. So what is this actually doing in terms of the distribution of phenotypes along the phylogeny? It could be that all these central populations are sharing alleles through, short distance migration that sort of homogenize them and keep them all similar. And so maybe these, because they are far away, are not part of this sort of gene flow pool, and that allows them to be different. Or it could also be that maybe all this gene flow between very, close in space populations is actually shaping the genome-wide variation. So even if, for example, the branching history of these populations did not look like convergent evolution, even if the other ones were sister to one another, gene flow between them and their striped neighbors could be, if prevalent enough, sort of erasing the signatures of past history and generating a new phylogeny in a way, or I guess patterns of genetic relatedness that we then use to build a tree and say this is convergent evolution, even if it might not be the case. And this is not a case that's usually discussed, although working at a butterfly lab, I sort of had it shoved into my face. Because Heliconius, the researchers actually think this is what happens in two of their mimetic rings, where you have, again, similar color patterns that are just junked in space, and if you sequence mitochondrial DNA or genes around the genome, you usually find that these different patterns segregate more by geography than their color patterns. But if you sequence the genes that have been over and over and over shown to make these color patterns, you find that they segregate by actual coloration. And furthermore, if you look at the ages of these alleles, they are way, way older than the geography says. So this could also be the case here, which led us to try to find the genes involved. This is very much a work in progress, so it's really messy. What we did was a divergence scan between these two species, mapped it back to a terrible reference genome that we're making. But we still found a peak in terms of allele frequency differences as well as FST. That peak has one gene in it. It's a very small scaffold because our genome is very fragmented, which is a dynein. And my first reaction was, what is a dynein? Right? So dyneins are proteins that move stuff around microtubules, which makes a lot of sense considering that in fish and amphibians, dark or light color patterns are made by aggregating or dispersing melanin granules in melanin-bearing cells. And this happens along microtubules, and it's been long known to be mediated by dyneins. So it makes sense. I've just waved my hands a lot. I will have 2 years worth of postdoc to actually test this. But if we assume that this is actually one of the genes involved in color pattern variation, then it would be really interesting to know what its history is. If you look at how these look, it could make sense. And I really wanted to show you something about the history of this dynein, but sadly that's in the sequencer right now. So for now, I can tell you that color pattern variation in these frogs in the is all over the phylogeny, but matter and the phylogeny matches geography. There is a lot of gene flow, which is definitely doing something, but we're not too clear what, and that there is a dynein associated with that. So we know a little. We're starting to find out what's going on with these guys. That I want to thank all my collaborators, money institutions, etc., and probably take half of this.